we go. We are talking today about DNA mutations, chromosomal mutations, and gene expression errors. So we're looking at when things go wrong. We've been talking in this unit about DNA and how incredibly precise and incredibly organized genetic information is, and it totally is. And you can worship God for his creativity and his precision in DNA. But we also live in a fallen world where there is sin and things break. And DNA breaks. And so when DNA is not expressed properly, we get all kinds of funny things. And we're going to be talking today about mutations and when things don't go right. So mutations is the name of the game. But we're not talking about, there we go. We're not talking about that kind of mutant. No, no, no. We're talking about this kind of mutant.
Oh my lord. Oh.
Like a four-legged chicken. So, yes.
Oh, that's so weird.
KFC would love it if that were more regular. They'd sell more toasters.
That's the other one.
That's cute.
So, take a moment, write that down. So, in your note-taking yesterday, when you were doing the gallery walk over chromosomal errors, you were looking at problems that happen when the wrong number of chromosomes are in a cell. So there were examples of having too many chromosomes and some things that were caused by that. There were examples of not having enough chromosomes. There were some examples where you had portions of a chromosome duplicated again. And so all of those things happen to the number of chromosomes in the cell, and that causes errors, causes mutation. Most of the time that results in the organism that has that DNA dying. Most of the time an embryo does not completely develop if its DNA is not good. That embryo will die. But sometimes it does make it to birth or to hatching or to growing a tree, whatever kind of critter it is that you're talking about. And so we will see variations and things that are different about that organism. And we call those mutations. Now, again, a note here about our view of the world as Christians versus the secular views of some other scientists. You will hear evolutionists say that the source of genetic variety out there, the reason there are differences from one individual to another, that natural selection can use to drive evolution and drive the change in species, is because of mutations. But if you look at mutations that we know of, almost every mutation, and the word almost doesn't really need to be there, with maybe only a handful of exceptions, every mutation that we are aware of is deleterious. It means it does something bad. It makes the organism less good, and something is wrong with that creature when there's a mutation. And so to say that mutation does good things and adds information to a system and adds complexity to a system makes about as much sense as saying that going through a book and highlighting sections and hitting delete makes it a better book. Only if you thought the book was long and boring, but it's not going to add more complexity, add more interest to the book by deleting portions. So mutations are downgrade, downhill things. And they are problems that are evidences of the fallen world. Okay, so that's what mutations are. What kinds of mutations are there? Translocation is one kind of mutation, where part of one chromosome breaks off and becomes incorporated into another part, or a part of another chromosome. So the nucleus of the cell knows that DNA comes in chromosomes and knows that DNA should be packaged in this particular way, and the cells even know how many chromosomes there are supposed to be in their organism. And so if a part of a chromosome breaks off, the cell doesn't just leave that piece of DNA floating around free in the nucleus. It says, no, 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 that's not okay. You're not a chromosome. You need to go into a chromosome. So it will take this piece of DNA and it will add it to a chromosome. Probably not the right chromosome. And so from then on, that DNA is misfiled. it's stuck in the wrong part of the DNA of that organism. And so a couple things could happen. This cell might go to try to read a gene, but the gene isn't where it belongs. And so the cell can't read that gene, and so consequently that gene doesn't get expressed, and something happens incorrectly in the organism. Or maybe that piece of DNA was added into the middle of a gene when the cell stuck that back into a chromosome. And so the cell is trying to read a gene, but in the middle of that gene, before it gets to a stop codon, it winds up reading more DNA than it was supposed to. And so it's now reading information for one protein when it's trying to make another, and the protein winds up too big, doesn't fold right, and the protein doesn't work. And so you'll see something happen incorrectly in the cell because of that. So translocation, you still have all the DNA, but it's not where it belongs, and it can't function properly. Okay? You can also have a deletion. Part of a chromosome breaks off and becomes lost. The part of the chromosome breaks off, the cell says, I don't know what to do with you, and so I'm going to either digest you, break you apart, I'm going to stick you outside the nucleus and let the cytoplasm deal with you, or the next time the cell divides, the chromosome that is missing that part doesn't copy properly in the next generation of the organism, doesn't have all the DNA it's supposed to have. So deletion is another option. So here we have all the information but not filed properly. Here we're missing some information. In both of those circumstances, we run into improper expressions of the DNA and things not looking right. So, oops, just kidding. Let me go back to where I belong. Where's my mouse? Oh, sad. What happened? Oh, there you go. There you go. What I meant to do was try to grab that over there. Okay. So this is an example of a translocation where a part of chromosome 4 and a part of chromosome 20 both broke off. And then the cell tries to fix it, but it gets it wrong and puts part of chromosome 4 on chromosome 20, part of chromosome 20 on chromosome 4. So what that means is that when the cell is looking for this information, it's going to go to chromosome 20 and look at the bottom of chromosome 20. Well, when it does that, it's going to be reading this information, not this information. And so it'll read the wrong thing, and it'll do the wrong thing because it's reading the wrong set of instructions. Okay? Another example. There you go. These are some examples of translocation errors. And they've done a cool thing with cells here, and they have added a dye to some DNA that will show where mutations are. So these cells have mutations, and you can see that at the cellular level. And then here are some mice where they have no musculature in the back legs. The back legs just kind of hang out. And the problem there is that the cell's information for how to make back leg muscles wasn't found. It went to where the DNA is, and it says, hey, here's where you know information for how to make muscles in the back leg. Open the book, and it's blank. There's no information there. Well, no muscles in the back leg. Okay, man, the mouse just gets born, dragging its hindquarters around. So that's an example of that kind of error. Okay. This is an example of a deletion where the code should have said CAT, but that A gets lost. It's a deletion mutation. And even though it's only one nucleotide that gets lost, now instead of reading CAT, now it reads CTC. And that codes for an entirely different amino acid. And from that point on, everything is going to be reading, is going to make the wrong amino acid because it's always read in groups of 3. So if you pull a nucleotide out, every group of 3 after that is wrong. So even though you only remove one nucleotide, everything in the gene after that is broken. So the gene will not function properly anymore. Okay? So deletion mutations are a problem, and they cause what's called frame shift mutations. Sometimes it's a big deal. Sometimes it's not a big deal. Here's an example. These are carnations, and the normal color for a carnation is this kind of pale yellow, where the dark yellow pigment is limited to the middle, and then as the petal gets further from the middle, it gets lighter. Well, this is a mutant, and here the entire petal made the dark yellow, and that's a point mutation. And you can see this doesn't kill the plant; it just looks different, right? So this is a mutation, and it is a loss of information. It's less information. This one is not only how do you make the pigment, but where do you put the pigment. Here, I know how to make the pigment, but I have no idea where to stick it, so I'm just going to put it everywhere, right? And so it's a loss of information that changes the color and the appearance of the flower. Okay, so there you go. Another kind of error is an inversion. I'll give you a second to write that down. Maybe more than a second. Okay, so we had translocations, we had deletions, now we have inversions. Inversion means to go upside down, right? Huli huli. And so this is a huli huli mutation, only they call it an inversion mutation, right? We have a part of a chromosome that got broken off, and when the cell says, I can't have you floating around in the nucleus by yourself, you need to be a part of the chromosome, it finds the right chromosome to reattach it to, but it sticks it on backwards. So now, instead of being read, left to right, it's backwards, and you would have to read it right to left, and the cell doesn't read that way. So this gene is going to be read backwards, and will probably not work when it's read backwards. Okay, and then a duplication. Part of a chromosome is copied twice during synthesis, and so the next generation of that cell, the next generation of that organism, has 2 or 3 copies of that gene instead of just 1, which makes that part of the instruction more active, and so the cell is doing that thing more than it should, which sometimes is not a problem, sometimes it is a problem, sometimes it kills it. And so duplication errors happen. And then point mutation is where we get one base pair wrong. And so that will create one wrong amino acid. Very frequently, this doesn't do anything bad for the cell. One amino acid wrong, the protein will probably still work. So this is the least problematic of the kinds of mutations that are out there, where you didn't remove an amino acid or a nucleotide, you just got the wrong one there. Okay. So inversions, duplications, and point mutations. Let me show you some examples of these. So here in a piece of DNA, that should be read TTA CCT, gets cut and flipped around and stuck back in. So now it reads TTA CTG, and you can see that that's a different amino acid big pair. And then the next codon should be GAC, but now the next codon is GGG, so that's a different amino acid too. And you can see that until you get back over here, it's going to be the wrong amino acid. So depending on how big that segment is that gets cut off and flipped around, the protein will be some degree of broken and will probably not do its job functionally. And so you will probably wind up with something wrong in the organism. Okay, so that's an example of an inversion mutation. A collie, what's wrong? Oh, so a collie should look like this, right? Dark black, dark brown, and a section of white. That's what a normal collie looks like. It's a tricolor. Here, we have a collie where it still has brown and it still has white. White is no pigment. But where the dog is trying to make black pigment, there's an inversion mutation, and the pigment doesn't work. The pigment doesn't color the hair. And so here we have a gray dog because there's a chemical being made and added to the hair, but instead of making the hair black, it's just kind of this weird gray color. So this is called a harlequin, and that's the name if you're a dog breeder. This is a harlequin color pattern, and that's the gray with a couple of spots of black, and that is considered an exotic color, and you can sell the dog for more if this happens because it looks neat, but it's a mutant, and the dog is broken, and the hair color just doesn't work right? So That's an example of an inversion mutation. A duplication. So now this piece of the chromosome was copied, but then the cell made a mistake and forgot that it already copied it and copied it again. So the new generation of this chromosome is twice as long. So all the information is there, but some of it's there twice. And I don't know if you've ever been editing a paper to turn in, if you were like cutting and pasting paragraphs back and forth to change where you talked about something, and you forgot to delete where it used to be, and now I, the teacher, am reading your paper, and you're making a lot of sense, and then I keep reading, I'm like, wait, you just said that. And I looked up, it's the exact same paragraph, and I'm like, oh gosh, it makes it crazy, and I take off points off the paper. Right? Same thing that happens here. The cell goes to try to read this chromosome and gets confused because it just read that. And why is this here again? And so things go wrong. Huh? Yeah, yeah. So here's an example of somebody's foot with a duplication mutation. So all of the information is there. Okay, don't make fun of these people. All the information is there, but there's too much information there. And so the body got confused when it was reading, what do I do with my toes, right? That chapter was confusing, and so it didn't know what to do with your toes, so it did that. And so all the DNA was there, but there was too much information, and the body got confused, and you wound up with hammer toes. Okay. Here, this is a point mutation, where it's supposed to be AGT-CGA, and instead, when the cell copies that DNA, it makes a mistake, and instead of putting a C there, it puts an A there. So now it's ATG-AGA, and you can see that that causes the codons to be read improperly, and you wind up with a different amino acid, but only one amino acid is different. The rest of the protein is the right thing, and so odds are it's still going to work, maybe just not quite as well. And then there's some examples there of frame shift, which we've already talked about, where if you take something out, everything after that is wrong. Okay, so this frog, poor frog, this frog has a point mutation, only one thing wrong with it. But it happens to be on a gene that tells the frog where to put an arm. Oh. Okay? So the frog knows how to make arms, but it doesn't know where to put arms. Where do I stick an arm? And so you wind up with a frog that has arms coming out of the wrong spot. And there's only one amino acid wrong. And it just happened to be a very, very important amino acid. Okay, so you wind up with funny looking frog. Yeah. Yeah. Okay. Go ahead and write this down. All right, we've already talked a bit about this beforehand, top of the lecture, but mutations remove information, remove precision, mutations go downhill with regard to organization. They are the exact opposite of evolution. Evolution requires uphill, more information, more complexity, more organization. Mutations, because of what they are, remove organization, remove complexity, remove precision. So to say that evolution is driven by mutations can't be true. Oftentimes mutations do nothing. You have, I'm sure, lots of mutations in your DNA that are different from mutations that your parents passed to you. You have your own brokenness. But oftentimes it doesn't have any effect. Your body's able to keep going, and generation to generation, these small changes add up. But in an individual, the errors that happen tend not to be a big deal. Only very, very seldom do they, show up and the creature all of a sudden dies. But that does happen. And even more seldom than that, almost never, is the mutation a good thing. And even if you were to look at examples where mutations, and we know that they're a mutation, and was good for the creature, even in those circumstances, it's a simplification of the creature. It's a lessening of the complexity that happened in that particular example to be good for the thing, to be less complex. But you're never adding complexity by doing mutations. So I've got, yes, a couple more slides. process. Okay, so when we talk about gene activation or gene deactivation, we're talking about, is the cell reading that gene right now? So every cell in your body has all the DNA that it takes to make you, and that is true of every organism. I could take a skin cell from Hannah, and with the right technology, I could grow another Hannah from a skin cell of Hannah, right? That's called cloning. And we're going to talk about that in a little bit here. So, but a skin cell does not need to function as everything in your body, right? Even though a skin cell has information in it to be a neuron, to be a kidney cell, to be a blood cell, to be a gamete, it has everything that it needs to do any job in your body. But it itself is only going to do the job of being a skin cell. So not every gene in every cell is read. Huge portions of the DNA in that cell have been switched off because it doesn't need to read that DNA. It still has the DNA, but it doesn't read the DNA. So the gene has been deactivated, right? And so that process of a cell saying, I need to read this, I don't need to read this. I need to know this, I don't need to know this. That process is called cell differentiation or cell specialization. So the cell has a job to do, and it needs to only read the information on your DNA that tells it how to do that job. And so certain genes are turned on or turned off. Picture like a circuit breaker. I don't know if you guys have ever been at home and you had too many electronic devices running and you popped a circuit, popped a breaker. Your dad probably was the one that went to the back of the closet and switched the switch back on. I don't know if you've ever done that yourself, or yeah, but wherever that is in your home, the breaker, when the breaker is off, no matter what is plugged into that circuit, it's not going to work, right? There are genes in the DNA of every organism. Oh, Keon. There are genes in the DNA of every organism that are like circuit breakers. And when that circuit gets turned off, no matter what genes there are associated with that circuit breaker, those genes are not read. And so your cells in your body and in every organism, not just your body, because there are any multicellular organism, there are jobs that the cell has to do. And it can turn genes on or off based on whether or not it needs to read that information. And there are switcher genes that the gene will say, you are currently in a kidney. You don't know or need to know how to be a heart, right? And so it turns off all the genes that have to do with hearts and turns on all the genes that have to do with kidneys. And we call those switch genes homeotic genes. And these are genes that are like circuit breakers. Okay, so if it gets switched on, that cell is going to do all that set of jobs and read that chapter of the DNA book in you. If that gene gets switched off, then all of the information in that chapter of the DNA book is going to be ignored and not read. Okay, so this is why, you know, normal frogs don't have arms growing out of their back, because the back knows it's not an arm. And so these homeotic genes turn on big pieces of DNA or turn off big pieces of DNA. Well, what happens if one of those genes is mutated? If the homeotic gene is mutated, then the homeotic gene turns on the wrong cell structures, turns on the wrong organismal features, right? So this diagram shows you regions of development, and it's the same for people and for flies, which is interesting. The same set of homeotic genes turn on the head region, turn on the upper head region, and the lower head region. So what does it take to be a brain? What does it take to be a face, right? It's the same genes in a fly. What does it take to be a brain? What does it take to be a face? Now, it's obviously different sets of genes because a fly's brain and your brain are way different things. But it's the same switch gene that says, I'm a head, I'm a face, right? And so, and a neck region is the same. And a thorax, and a lower thorax, and an abdomen, and a lower trunk, right? And then the reproductive structures. So all of this It's the same sections of DNA. Now the code obviously very different for a fly than from you. I'm not saying that you're flies. I'm not saying that you're evolved from flies. I'm just saying that when God wrote the switches, he wrote the same switches for a fly and for you. And it's the same homeotic genes that get switched on or switched off to tell that cell, this is the part of the body that I'm in. Okay? Which is why your legs don't try to grow a brain, because your brain is not there. Okay? Even though your legs have the information to make a brain, they don't try to. Okay? So this is, it's trying to show a half male, half female, to show you that it works, right? It's not very sad if this were an individual. Right? Yeah, okay. So what happens when this goes wrong? What happens when this goes wrong? What happens when this fruit fly had switches work for I'm the front of the head, I'm the lower part of the head, I'm the neck, I'm the upper thorax. But instead of having the I'm a lower thorax, it had another switch that was thrown and said I'm another upper thorax. So it had two sets of wings, right? And a normal fruit fly should have had different structures here, but the wrong genes got turned on. Okay?
It's still flying.
Yeah.
Whoa.
Yep. So,
oh gosh, not that picture.
now, this is really interesting. So, this I've already shown you. This I've already shown you. These are honeybees, and this is a honeybee that has had a part of its body grow wings that shouldn't have grown wings. Now, this is a rat, and when this rat was a single cell, the human genes for how to make an ear were added to that single cell. Okay. Now, unless the rat had been told that some part of it was a human ear, then those genes would have just been in the rat and you would have never been able to tell. But when the rat was a couple of cells big, so in one cell, they added the DNA for a human ear. And then when the rat was like 16 cells big, so it had developed a couple of times, they tricked one of those 16 cells in the 16 cell stage of the rat to say, make a human ear. And they turned on that set of codes. Now the rat develops, and the rest of the rat is fine, except that it has a human ear on its back. Now, this was done to prove that we could grow human spare parts on other animals.
How is that possible?
So it has all the parts of an ear.
Well, I think in this particular example it's just the external structures, but it was just done to show that we could do it. We don't do this yet, but it was to demonstrate the technology that if some soldier gets his ear shot off, instead of making a plastic ear and trying to make it look like his face, we could, if he's got a couple of weeks, we could grow one for him on some creature and then transplant that onto the guy. And if we use his DNA to tell the rat how to make an ear, then that would be his ear, grown on the back of a rat. Well, and I think in this example it was just the external structures. But it was just a proof text to show that we could do it. We're not doing it yet, but it was just somebody who said, look what I can do. So this happened in nature. Okay, this is a turtle. And the turtle had two parts of its embryo with the switches thrown for make a head. So the turtle has two heads. And you'll see that more often in reptiles than other kinds of creatures. Reptiles, for some reason, that grow a head switch gets thrown too often sometimes. So you'll see two-headed snakes in the wild. You'll see two-headed turtles in the wild. And both heads of this turtle are functional heads, right? Because once the cell was told, make a head, it did, and all the structures were there. So, turtles I don't know how intelligent they are, but it could argue with itself. Which way am I going to go? I'm going this way. No, I'm going this way. Shut up! I'm going this way. Right? So this happened in nature. Here, here's a pig with two faces. Okay. It's only got one brain and it has two eyes, but it has two noses and two mouths. A pig already eats a lot. I can't imagine what a pig would do if it had two mouths. Right? It only has one stomach. But yeah. That's it. Your dog has two mouths? Okay. So another time that you see genes turned on or off is not just because my job as a cell in this part of the body is to do only this thing, but you also see genes turned on or off during development. So genes can be active during a certain part of development and then be turned off in other stages of development, and that function goes away, right? Or that function is created if the genes are turned on at some point in the process. So sometimes we call these control factors. A control factor gene controls when a cell expresses a particular trait, and this has to do with development. So some examples of when this goes wrong. Here we have a freakishly tall man. Okay, this guy has a problem called gigantism. Okay, gigantism. His DNA is the same as of his other DNA. I don't know which one of these guys is family members, but he's getting married. Got to imagine some of these people are family, right? So his dad is not freakishly tall. The reason he's freakishly tall is because the gene that creates human growth factor that causes you to grow and the gene that turns off your growth plates in your bones didn't work. So he keeps growing. So the average man stops getting taller at about 20, 21 years old. At that point, you're as tall as you're going to be. And your growth plates fuse, and your skeleton won't get any taller. So if you take human growth hormone as an adult man, you don't get taller. You can grow more soft tissue, so you can get more muscular, but you won't get any taller. This guy, his growth plates never fused, and his human growth factor never stopped getting produced. So yeah, so he will grow until he's too big for his heart to keep alive, and he will not live very long. Okay. If you guys ever followed pro wrestling, it's been a while, but Andre the Giant, you guys familiar with him? Have you heard of him? Another guy with gigantism. So Andre the Giant was much taller than the average dude, but his growth plates eventually did fuse, so he stopped getting taller after he was already too tall. But he kept making human growth factor, so his soft tissue kept growing, which is why he was so stinking big, right? He was like a marble. And it's not because he was obese, but it's because his body kept saying, I have to keep growing more of Andre. So he's going this way, but it can't go up. So yeah, and eventually he died of a heart attack at like 45 or something like that, because his heart couldn't pump the blood through that much body. So this guy's getting married, and I hope he's got a couple of good years of happiness, because eventually he will grow himself to death. Okay? So
I heard that they eat something.
yeah, oftentimes the skeleton isn't strong enough to support itself too. So that's one example of that. Okay. Is that the last one? I thought I had another one. I guess that's it. So cancer is another example, right? Where a cell says, I'm going to keep growing right now. And the body says, I don't need you to keep growing. Oh, I'm growing. And all of a sudden that's a tumor, right? And so it starts sucking life resources from the tissues around it and makes the person sick, and it can eventually kill you. There's one other really interesting example that I initially wasn't going to show you, so I didn't put it in the lecture, but I'm going to. You have human development. At one stage in the human embryo, there is a tail.
We have a tail?
Yeah.
Right? Humans have tails
What?
as an embryo. Inside your mom's womb, there's a stage where you have a tail. Well, at some point, there are genes that are switched on that absorb the tail. Okay? And so before you're born, your tail has been absorbed. And all the bones that used to be in your tail get really small and fuse together, and we call it your tailbone. Right, okay. But if that set of genes to absorb the tail don't get turned on, then you are born with a tail, right? So,
No way.
yeah, so I want to show you some pictures of humans with tails.
Oh, what?
No way, man.
What?